Hi, my name is Nihal Singh, and this is my reproductive system portfolio presentation. I'm going to be covering the regulation of sperm maturation in the germ epithelium. So, we have a process called spermatogenesis, which is the process of producing male gametes or spermatozoa or sperm cells from undifferentiated germ cells called spermatogonia. So spermatogenesis is basically the process of spermatogonia turning into spermatozoa or sperm cells. This is going to occur in the seminiferous tubules of the testes. It's going to take about 64 days, and 100 million or more sperm per day are produced. It's it takes it has three stages: mitotic proliferation, meiosis, and packaging. And we're going to get into what those are. So, mitotic proliferation is the continuous mitosis of diploid spermatogonia in the outermost layers of the seminiferous tubules. So basically, mitosis is like an exact copy of the original cell. So two identical daughter cells that are copies of the original cell. So we start with the spermatogonium that's going to undergo mitosis to become spermatogonia. Then there's another round of mitosis for the primary spermatocytes to form. Then we have meiosis. So meiosis is a little different than mitosis. It is not identical daughter cells. There are 4 daughter cells produced because there are 2 rounds of meiosis, and they are genetically unique in as compared to the original cell that undergoes meiosis. So the difference between mitosis and meiosis is that mitosis is not genetically unique from the original cell. Meiosis is. So with meiosis, the primary spermatocytes are going to undergo the round of meiosis to become secondary spermatocytes, and then another round of meiosis to become the spermatids. And from there, we undergo packaging. So the cytozoa and organelles are removed, and the head, midpiece, and tail are formed to produce the mature spermatozoan or the sperm cells. So the spermatid becomes the spermatozoan. And here we have a picture of where that's happening. That's in the seminiferous tubules. So it's going to be moving in this direction. So the spermatogonium is going to start here, and then it's going to go through that whole process that we just described, the spermatogenesis process, and then it's going to become the spermatozoa once it gets towards the lumen side of the seminiferous tubules. Now, we also have Sertoli cells, which support the sperm cells. So as you can see in this image right here, this is the spermatogonium, this is the spermatogonia, and it's kind of going through that process that we just described, spermatogonia to the primary spermatocytes, secondary spermatocytes, spermatids, and then finally the spermatozoa. So the Sertoli cells are supportive cells towards the sperm cells. So they provide nutrients, protection, fluid, a barrier to prevent pathogen entry, and hormone support. So this image right here kind of shows an overview of that spermatogenesis process that we were talking about. So we have the spermatogonium that undergoes mitosis. So it's going to be an identical copy, so two identical daughter cells, and they start off diploid, um, meaning two pairs of chromosomes, so two pairs of 23 chromosomes. Um, After the spermatogonium becomes spermatogonia, they undergo another round of mitosis, and two more um, daughter cells, identical daughter cells, are formed. Then there is another round of mitosis, and then the spermatocytes are formed. So with mitosis, it's going to be staying diploid. Now, when we get to meiosis, so we have the first round of meiosis, and they're going to become the secondary spermatocytes, and now they are haploid. They each have one strand of the 23 chromosomes instead of a pair of 23 chromosomes, so 23 chromosomes in total instead of 46. Then they're going to go undergo the second round of meiosis and become the spermatids. And after that, they're going to undergo packaging or spermiogenesis and become the sperm cells or spermatozoa. So the spermatozoa is displayed over here. We have the head, the midpiece, and the tail. We have the head is kind of covered in an acrosome, which is an enzyme-filled cap that allows drilling into the ovum for fertilization. We have the midpiece that's concentrated with mitochondria, and the tail, which is flagellum with microtubules for movement. So that aids in the movement of the sperm towards the egg. All right, so I have a worksheet to review what we just talked about. So if we're going to put the stages of sperm maturation in the right order, so we start off with the spermatogonium, then it underwent a round of mitosis to become a spermatogonia. Then it underwent another round of mitosis to become the primary spermatocytes. After that, it becomes it undergoes a round of the first round of meiosis, becomes the secondary spermatocytes. Then another round of meiosis to become the spermatids. That is going to undergo packaging, becomes the spermatozoa. So the right order for this would be C, D, F, A, and E. Sorry, A, B, and E. Secondary spermatocytes, spermatids to spermatozoa. So C, D, F, A, B, E. All right, next one, we have some terms, and we need to match them to the appropriate definition. So mitotic proliferation, you'll remember, was it remained diploid when we did those rounds of mitosis. So we have this right here. This is continuous mitosis of diploid spermatogonia in the outermost layers. So that's going to be B for number one. Now meiosis, that went from diploid to haploid for that first round of meiosis. And there was two rounds of the division to produce haploid spermatids. So that's A. And packaging. That was the spermatids to the spermatozoon. That is C. Cytozoals and organelles removed, head, midpiece, and tail formed to produce mature spermatozoon. Number 3, Sertoli cells provide nutrients, protection, fluid, blood testes barrier, hormone support, or all of the above. If you recall from the slide, it is all of the above. Sertoli cells provide all of these. Number 4, spermatogenesis is the process of producing male gametes, spermatozoa, from undifferentiated cells, spermatogonia. That's correct. It undergoes the process of going from spermatogonium to spermatogonia to primary spermatocytes, secondary spermatocytes, to the spermatids, to finally the spermatozoa or the sperm cells. So that is true. Number 5, the tail of the spermatozoon is covered in acrosome. That's not true. That's false. The head of the spermatozoon is covered in acrosome. And number 6, we're going to match the term with appropriate definition. So we have different types of sexes that we can identify. So the genetic sex has to do with the chromosomes. So that's going to be XY and XX. So that's how you determine the genetic sex. The gonadal sex is going to have to do with the testes and ovaries. Depending on someone has testes or ovaries, that determines the gonadal sex. Phenotypic sex is external genitalia and reproductive tract. So genetic sex, so A is going to be XY and XX. Gonadal sex is going to be testes and ovaries. Phenotypic sex is going to be reproductive tract and external genitalia. That concludes my presentation on the reproductive system and the regulation of sperm maturation in the germ epithelium. Thank you.